So today's talk, I'll mainly discuss about the mutations, mainly the disease-causing mutations in globular membrane proteins using machine learning techniques. How to identify these disease-causing mutations. We have been asked to discuss much more details about this mutation level and the machine learning. Okay, so I'll discuss various aspects, and I'll tell you what we learned from these disease-causing mutations in proteins. Okay, I belong to the Department of Biotechnology from IIT Madras, and it is founded in 2004. Currently, we have 31 plus recently 4 joined to 35 faculty members and more than 600 students. And we offer dual degree, both engineering and the sciences, that is BTech, ComTech or BSc MS. And we also have the degree programs like the MS by Research and PhD degree programs. Okay, so we are focusing on 4 different aspects: bioprocess engineering, biomolecular sciences, computational biology, and medical biotechnology. And computational biology, we have a strong group in our department. We are focusing on various aspects such as structural bioinformatics, comparative genomics, molecular biophysics, neuroscience, as well as structure-based drug design. Okay, we have several PhD positions available, right? Even the UG students with a clear gate, right? You can apply for PhD. You can do direct either MS or you can do direct PhD, MS and PhD together, right? I was get a clear gate, and then you apply, and then we have interview procedures, and there are several opportunities for both MS as well as PhD. Fine. So here I show the works what we are doing in our lab. Actually, I'm heading this protein bioinformatics lab. Okay, here you can see the. Let me see. So here, this is our logo, right? Protein bioinformatics lab, actually designed by my students, right? Here you can see the DNA is here, and you can see the proteins here. And we do statistical analysis and machine learning techniques. Okay, this is your statistical techniques. And okay, this also we work on this human proteome and all. So here there is, you can see a man here. So he is be happy to do this type of analysis. Okay, there is a background of designing our lab logo. So mainly designed by my students. Okay, so these are the 2 books. Okay, the protein bioinformatics and protein interactions, right? This one I published in 2010, this in 2020. Right? So you get almost all the details of this machine learning techniques and various aspects of protein folding, stability, as well as interactions. Okay, so I'll briefly tell about what type of work we are doing in our lab. So maybe if you are interested for a project, or if you are interested to do your MS or PhD or some other various proposals, right? This will be helpful. So we focus on majorly 2 different aspects. One is to understand the protein structure and function, and the second aspect is for the structure-based drug design. So how to use this information, what we obtain in these studies, right, into the applications point of view. So in protein interactions, right, so we focus on protein-protein interactions, protein nucleic acid interactions, and protein ligand interactions, and protein carbohydrate interactions as well. So we derive databases. Okay, if you look into my website, you can see several databases called Approximate, ProCap, ProNav. These are the databases mainly for the protein complexes, right? And these are the papers. So we publish these works. And once you develop a database using the data available in these databases, right, we can derive some parameters. We can derive some features, and you can relate what features are important to understand this binding affinity of these complexes. Right? So we do a statistical analysis. Finally, we develop algorithms for predicting the binding affinity upon mutation. Okay, so this we do it for the interaction. On protein stability, we developed this 2 databases. One is for the globular proteins, ProthamDB, and MBTHAM for the membrane proteins. These databases received more than 1,000 citations. So it is very popular in the literature. And we use these databases for developing computational tools. Not only in our lab, several labs in the world, they are using our databases for developing tools, right, for predicting the stability upon mutation. Then we also try to see what factors which makes some proteins very stable, right? Hyper stable, like more than 100 degrees. right? So what the different organisms, some organisms they grow in 37 degrees, some of them they stand up to 89 degrees. And what makes these proteins to survive for more time? And it is important to enhance the temperature so that it can be active, right, for some more time. So with that, we discussed the analysis, we derived it, and then we found that some factors like the mainly hydrophobic interactions are very important for enhancing the stability of the thermophilic proteins. Then we did on protein aggregation, that some proteins aggregate. So aggregation is both positive and negative aspects. Some of them they good for promoting folding. Some of them are causing diseases, and also it's useful for developing nanomaterials. So we developed a database for aggregating peptides and the proteins, right? And from that, if you give the sequence, it will tell you whether it is aggregating or not. Or if you give a protein sequence, it will tell you where which regions are aggregating. Right? That's where the program it's predict at very high accuracy. Then another aspect is how fast your protein can aggregate, or how slow a protein can aggregate, or any peptide can aggregate. But that also we have methods to predict these things, and you can see much more details right in my papers or in my website. So these are the basics. Okay, then we go to the application point of view, right? So here we use the disease causing mutation. This is what mainly today I'm going to discuss about the disease causing mutation. Okay, what are the various databases, what are the various tools, and what are the cancer hotspot residues. Okay, also we do some RNA sequence analysis, right, for the neurodegenerative diseases like Alzheimer or Parkinson or the Huntington and so on. So then that's I'll give you more details right in this talk. So we are working on structure-based drug design to design lead compounds of the BCL2, and this is our target for the cancer. So in this case, we design the molecules, right? So and then we use this cover hopping, and we synthesize, and then we purchase some of the compounds, and we did all the biochemical experiments. We found that some of the compounds identified in our in silico approaches are better than the other normal compounds, right? The control data in the literature. And then we went for the animal studies. Actually, we gave this compound to we injected these rats with the tumor, and in 2-3 days you can see the huge growth of tumor. And we gave our compounds, and it reduced significantly, right? And then after a few days, the one which we did not give the compounds that have enormous growth of tumor, what we gave this compound that reduced very significantly. Okay, then also we design inhibitors for the dengue or the other diseases, and for also the potential compounds for this cancer using this colorectal cancer using CS kinase as the targets. Okay, in COVID-19, right? Yes, so we did some of the analysis on COVID-19. So some of the aspects include the synergism effects on drug repurposing, and the structural analysis of spike protein and the antibody complexes, right? And we propose some antibody repurposing approach to identify the antibodies. And if you have these antibodies in our bodies, and then then after we did the mutation studies, what will happen if we mutate? Now the Omicron variant, you can see that the variants, right? So what will happen if you mutate that particular residue in that RNA binding domain, right? How they increase or decrease the affinity or increase or decrease stability, and how to interpret, okay, why this is the highly infectivity rate is very high and so on. So we can we did the analysis as well as the epidemiological models we have developed. Okay, so how the people affected with this COVID during stages with the lockdown and other social distancing and so on. Right? So we did that analysis on COVID-19. But these are the main focus what our lab is focusing on, right? So now I go to this main topic. Right, fine. Now, most of them are you heard about AI? So I need to have some sort of discussions. So I ask questions. Either you unmute your mic and tell the answer, or just you can write your answer in the chat box. Right? Both are fine for me. Right. So how many of you are aware of AI? Anybody is aware of AI? What is AI? Can someone answer the question? Are you aware of AI? The answer is yes or no. So I need one of the answers to start with. Yes, no answers? Okay, good. Yeah. So AI is artificial intelligence, right? So are you aware of the applications of the AI? Because now everybody talking about AI. Because if you talk about the computer science, or if you talk about the bioinformatics, or if you talk about the computer biology, right? So you can see potential application. There are so much applications using AI. Recently, if you see the protein folding problem, so predicting the 3D structure of a protein from its amino acid sequence, it's long-standing issues in computational biology. So using this AlphaFold, so they use the AI-based technique to capture the information from existing structures and then develop this AI-based method to predict these structures of several proteins. Right? So even the complete human proteome, right? So 21,500 proteins, right? They completely they predicted the structures and developed a database. Right? So AI has a lot of applications, right? In different aspects. Right? This is one side. Another side, if you see, they are talking about big data. That if you see the last admissions in the Indian universities, most of the computer science they wanted to have the specialization of either AI or big data, right? All the students they have the specialization in AI and big data because that is having a lot of future in life, right? So we talk about the big data. So it started 2 decades ago in physics, like mainly astronomers and high-end physicists. Right? So mainly the particle collision events in CERN, right? So the data are very huge. There is more than 15 petabytes, right? There is about 15 bytes. So they use this big data. Right? Now the computational biology, the biologists also joined, right, in this group, right? So based on the recent data which are available at large scale, right? You can see we have the data for the genomics, and we have the proteomics, and we have the transcriptomics, and we have metabolomics, right? We have different types of omics data. Because of this omics data, so obtained from these next-gen sequencing techniques, right? Life science also entered into, right, the group of this big data analysis. Okay, so even if you talk about the COVID, right? So we have more than 1 million genomes which are sequenced, right? That's a few months ago. So they got the sequences of several COVID infected patients, and they compared to healthy individuals, and they see why we have the mutations and what are the various problems, right, in this in this protein, right? So you can see that. So they try to understand what are the various mutations, which mutations are causing disease, which mutations are neutral, and so on. This is very important, even the current scenario. Right? So to have entered into this big data, the various initiatives, right, which have been taken, right, by the various institutions, right, worldwide. Right? So I explained some of these few aspects. One is NIH, that is Institute of Health in the US. Right? They started the BD2K, that is right. So they wanted to have this change big data to knowledge. Right? This is what they put BD2K. So there are 12 different centers to share the research objects, right? So these objects, I mean, this is the collection of resources, storing the data and computing and developing algorithms and all the aspects, right? They like to share the data, right, and collect the data at large scale. Then we completed 1,000 genomes project that is 10-15 years ago. So now we are having the 100,000 genome project that also now is almost done. So collecting the various data for the genomics, right, and analyzing data. Currently, due to COVID-19, we have plenty of data, right? Millions of data are available now, right? So having to analyze the variants that in this the COVID-affected patients. So now to analyze the genomic data, there are about 1 million individuals they target initially, the US Precision Medicine Initiative and the Beijing Genomics Institute in China. So try to analyze this genomic data, right, for various diseases. In India, so here also maybe 2 decades ago we started the OSCARD, that is the open source consortium for computer-aided drug delivery, right, in India. So here what they do, so we are collecting all the resources, mainly the programs which you develop, right, across India. So made it a common platform to put everything there. In this case, you don't have to write a program again and again. So if you want to calculate some of the features, it's already available, right? You can just use that program for your analysis, just to design your problem, and the features you can collect, right, from this consortium. So your website and you can use that. Currently, the Department of Science and Technology and the Department of Biotechnology, Government of India, so they are taking efforts to collect all the resources, right, even the databases or the web servers, the tools, everything, and try to make a capital. So in this case, And if you want to use any of these portals, you can use the data and you can use servers and you can use all these computing resources which are developed in India. Okay, fine. Okay. So now what are the various challenges? There are various challenges in big data in biomedicine. Firstly, we need to create analytical tools because we're having this huge amount of data and solving scientific puzzles, right? And analyzing millions of gigabytes of data. And basically, main aspect is the overcoming the barriers to access patients' health records because patients' health records are confidential, so we cannot reveal it to anyone. So we need to do this encrypt and encoding and decoding, right? So to access this data and how to use that, right? So then to address these issues, there are several databases that which have been developed, and they have the subset of databases, right? In each case, they rather than having the full data size, because if you take the protein data bank, they get a lot of information, but mainly the research is used with coordinates, XYZ coordinates. So a subset of data, in this case, we can avoid the usage of enormous space and you can have a subset of data, so which are highly used by the researchers. In this case, we can use fairly and efficiently this data. Then we have the developments, the high protein research and the big data to healthcare, because now the healthcare, the AI and big data are playing an important role. And if you see a few years ago, there's a paper published in Nature. So they use AI-based method to discriminate between the skin cancer as well as the normal ones using AI techniques. And the performance of this AI-based method is better than the dermatologist. They also sometimes wrongly diagnose, right? So here the performance of discriminating the malignant as the normal one, neutral ones is very high. So now this needs bioinformatics scientists. They look for people, those who are familiar in life sciences as well as statistics and computer programming. There's currently huge amount of data available using the transcriptomic data or the genome data or the whole genome data or proteome data. So it is very essential to analyze this data, right, for the healthy individuals as well as for the patients, right, to obtain information as well as how the response to medicine. Right? Because personalized medicine is very important because the response for some patients are good in some medicines, but not for all other medicines. Right? So now give the medicine and get the output and then see how it works. And based on that, they develop AI-based methods, right, or machine learning-based methods to see, okay, these are the symptoms. Okay, this medicine if you give, this could be the possible right medicine for this type of patients. Right? You can do that. So if you see COVID-19, you can see a lot of advancements in COVID-19 using the machine learning technique. They try to understand. Okay, so for example, if you see this COVID-19, so the virus, attacking with this human protein, like the spike protein, the virus, and the ACE2 in the human, where they transfer the information, right, spreading this infection. So now see the mutations and what are the possible mutations, and some mutations happen more than 1,000,000 times. Some mutations are very rare, right? So some of them are causing disease, some of them are neutral. So you need to analyze all these details, right, in almost all these diseases, including this COVID-19. Fine. So now with this background, okay, I'll start my talk. Right. So first, we need to know the mutation. So what do you mean by mutation? How many of you are aware of mutation? So what is definition for a mutation? Either you may write in the chat box or you can unmute your mic and you can answer. This will help me to go into depth or just I can go to the advanced level. So what is the meaning of mutation? Anybody is aware of mutation? Good. So what, either to take the DNA change or the protein change, and it's good. Yeah, it is change. There are different types of mutations. So, for example, if you have this is your okay, I'll take the DNA sequence CATC. Okay. So then I give one sequence, right? Okay, A, C, A, T, C. So can you see a mutation here? So you can, here this mutation is, you can see the second position, T is mutated to C, right? So in this case, this is called the mutation, right? So now if you see, there are different other aspects, right? One is the insertion and some also deletions. For example, if it is AT, okay. So now in this case, so the C is inserted here, this is called insertion. And if this is the case, and then okay. So in this case, A is here, but here there is completely not there. So in this case, there is called deletion. So either you can see insertion or the deletion, right? Or you can see the change in amino acid residue, right? Substitution. So there are three different types. Okay, this you can do in the DNA sequence, or you can do in the protein sequence as well. Protein sequence we call as synonymous mutation, non-synonymous mutation, depending upon whether they change in nucleotide, whether they change the amino acid or not. Okay. Okay. So now in the protein, they're replacing one amino acid to another amino acid residue, right? For example, if you change this alanine to valine, right? How many amino acid residues, naturally occurring amino acid residues in a protein? How many naturally occurring amino acid residues in a protein? I get 80% of the participants are biologists. If you write the answer, I can go fast. How many naturally occurring amino acid residues in a protein? 24. Any other number? 20. Okay. Any other number? 22. Okay. Yeah. So there are 20 occurring amino acid residues, right? So they are mainly grouped in two groups. One is hydrophobic residues and hydrophilic residues, right? So if it okay, actually I prepared my slides as an advanced level. That is why I don't have these basic slides. Okay. So if you take the hydrophobic residues, you can see the aliphatic groups, right? Aromatic residues and sulfur containing residues. So for example, can you tell a few aliphatic residues? Because this information are required when I go forward with this mutation analysis. Otherwise, difficult to understand. So there are a few aliphatic residues. Aliphatic residues contain CH2 groups. Right? Can you name a few aliphatic residues? For example, leucine, valine, isoleucine, so on. And there are a few aromatic residues. Can you list few aromatic residues? Aromatic residues contain rings. Do you remember any aromatic residues? Any answer is fine, even if it is not correct, it doesn't matter. Right? Good. Phenylalanine tryptophan is very good. So there are three aromatic residues. And if you have the polar residue, there are three different groups. Some of them are charged, positive charge, negative charge, like the glutamic acid and aspartic acid, negative charge, lysine, arginine, positive charge, and so on. Okay. Good. Yeah, fine. So now in the replacement of any specific residue, right, to another residue, it changes the structure, it may alter the function, or some of them cause diseases. Okay, that is what is happening even in COVID-19. Right. So I put some of the amino acids here. These are the aliphatic residues, right? Alanine, valine, leucine, and isoleucine. If you see, alanine is small one, right? And the valine is the little bit bigger one. So leucine and isoleucine, that is bigger than the alanine, valine, right? You think about the constitution in your protein. Okay, if this is these are the residues. Okay, so here this is alanine is here. Okay, this is only this much space is available. Say this A. If this A is changed to L, what will happen? this L, the L has space to accommodate this at the place of A. Right? Because this is how the protein looks like, and A is here. This is your A. Right? If you change A to L, can you accommodate this L to A? Yes or no? Based on size, because both the physicochemical properties both are same, both are aliphatic residue. So we have very less space here. So L, the A is here. Now if we change to L, what will happen? Can you accommodate L in this place? Replace A with L. Yes, what's your answer? I need some output. Yeah. I need some input from you. Yes or no? Can we replace this L A by L? Is it easy to do it or not? We don't have space. This is A is very the only this much space available. So accommodate A. If we mutate A to L, okay, so because the L is a larger one, so we cannot put it here. So in this case, this will destabilize the protein, right? So it changes this function. Okay. So there is another example that is called the hemoglobin glutamic acid 6-valent. So glutamic acid is a negative charge amino acid residue, right? So when you mutate to valine, valine is a hydrophobic residue, right? It forms fibrils, right? And this is block this oxygen transport. That is why it causes the sickle cell anemia. Right, this hemoglobin is a transport protein, right, in human body, right? It causes this have the breathing issues. Like as you can see the P53, this is the tumor causing protein, right? It interact with the DNA, right? If you have some mutations, then interface, right, it changes this affinity. Then it's also having several mutations. It's very arginine 248 in this protein that is very commonly occurring in many cancer patients. Okay, so now some of the mutations, right, these changes of the amino acid residues would cause disease. These called the driver mutations or the tumor causing mutations. And some mutations, the changing one amino acid to another amino acid residue, so that is not having any effects on this tumor growth. So these mutations we call as a passenger or these called neutral mutations. And this understanding this driver mutation, the passenger mutation is very important for the development of any therapeutic strategies, because depending upon the mutation, some may or may not respond to it, right? Okay, so I'll discuss mainly about the databases available in the literature and how to do this analysis, and finally we'll see whether we develop algorithm, right, to predict these mutations or not. Okay, so the biology experiments, they produce some data. So they produce data about the sequences and the structures and expression profiles, pathways, and so on. Right? So most data which are available scarcely in the literature. So they get the data and they publish the data in the literature. It is very randomly available. It is very scarcely available because they publish and then distribute. So it is very important to collect the scattered data and together and then put in a proper order, like a form of a database. So database you can define as an organized collection of the information in computer readable form. So here and there, if it's available in the literature, you collect all the information and put in the form of a database, right? Okay. So for example, if you want to develop a database, what are the various factors you think about to develop a database? Okay. So here there are various aspects we think about. So I'm sure all of you are using is one of the other ways databases. So everybody is using Google. Most of them are using WhatsApp. Right? Many various ways you are using database. For example, if you want to develop a database, or when you want to use a database, right? What are various aspects you think about? What do you need when you develop a database? Can you tell me some point? There are more than 100 points you can tell. Can you tell some points, right, which are very important when you have a database? Either you look for a database or you develop a database. Can you tell some ways what are the various aspects one has to consider to develop a database? So anybody is using Google? Nobody is using Google. We want to get information, any information. Okay. So if you Google if you search the information what you need, so how long you will wait? So you will wait. You will how many minutes you will wait to get the information? Okay. So when you make a database, right, there are various aspects we need to consider. One is the data should be authentic. Right. Currently, if you see there are so many WhatsApp messages, right, you can get, but most of them are not true, right? But when you have a database, for example, we take the NCBI, right, or the Protein Data Bank, or Unipro database, right? So all these databases which are authentic information. So you can use the database, right, which are provide accurate information, right, and you can trust the data which are available in these databases. Likewise, if you use any database, the database should be authentic, database should be informative, database should provide the correct information. And the database, right, should be accessible all the time. If you go to any database and if you are not able to access, right, 2 times you try or 3 times you try, if you don't get the data, so then after that people will not use it. So it is available all the times, and that should be fast enough because now nobody is having time to wait. So even if you Google, right, you want to expect the data right in seconds, like or less than a second, right? If it takes long time, 10 minutes to retrieve the information, then you don't use the database. Right? So you look for the database because there are so many options, right? So many databases, right, available in the literature. So in this case, you don't use the database, right? So there are various, and then the database, right, should be redundant, and database should have the information, right, what you want to get it, right, and should have the search options so that you can easily get the data right from this database. Various aspects, right? Okay. So because you are not talking, then I'll go ahead. Fine. So if you talk about the characteristics of database, right, it follow different characteristics. First one, the contents. Contents is very important. What do you put in your database? When you develop database, what are the data you want to put in? Then also you think about who will use your data. Because if you have a database, right, now we see people are selling the database information with just huge money, right? Because information now is equivalent to money, right? So the contents, what if you develop a database, what are the information you can put in your database? That is very important. Then who will use the data? That is also very important. The contents is very important. Then the second thing is, if you want to develop a database, you are familiar with various aspects, but not all are familiar with this your terms. So we need to have this ontology. That means we have the valid terms and definitions. Whatever the terms we use, for example, if you use delta G, right? So those who are familiar with delta G, right? So you develop a database, you know what's delta G, but not everyone. So that is a free energy change. So for all the cases, you need to write the terms as well as the definitions. Then for making any database, right, we need to have a logical structure. What is your major aspect? What is your major database? What do you want to put in? And what are the relationship with other information in the literature? This is called a schema. So you need to have a logical structure or expression of the interrelationship of the information what you are provide in your database, right? That is called schema. That is very important. Then for each data, you should have a format. If you see the Uniprot or if you have the PDB, so they follow a specific format. First one is a compound, header, and then you have the sequence, and then you can the structures, coordinates, atom, record, everything, right? So they have a specific format in all these standard databases. So you need to follow a specific format. Then you have the routes to select the retrieval of data. For example, if you put so much of data in your database, you should provide options to retrieve data. Otherwise, it's a big mess because you put a lot of information, right? So you have the routes to retrieve the data and you present the results, right? And you have provide the download data so that you can use it for the analysis, right? So nobody want to repeat the same again and again. So you have the downloadable data which are ready to use, right? Even everything is people don't use ready to use, ready to eat, right? So if the data are ready to use, then they will be popular, and many researchers they like to use your data. Then we need to give to other databases, right? Because if your data is somewhat otherwise, it is related to other databases available in literature, you need to give the proper information so that if they want to have more information, get that information from other databases, and then they can get additional data for interpretation of results, specifically if you have the original publications, right, of the where you get the data, right? I'll show the examples. Okay, here this is your contents. So this is the database I developed a few years ago. That's called Protham database, right? Currently, it is widely popular used by protein researchers and received more than 1,000 citations and as well, right? So here we have the data. So we have the protein name and the source and the PDB ID, uniprot ID, and the secondary structure and the uh, stability data and the mutation data and everything, right? They have this database. This is ontology. So these are the various terms I use in this database, and I have all the explanation for all the terms I used in this database. So number is entry number, enzyme commission number, PDB wild is protein data bank for this native protein, and so on. So we have the schema. So here this is my database. My database is experimental database. All that I collect, these are the experimental data I collect, the thermodynamic data, or the methods and conditions, or the literature, everything put here in this the program database. Then I give the link to other databases like the sequence or structure and the PDB data and the motif data, everything I put together in this database. I put the arrow here and put everything in this arrow. Then I give a search option. We can use the search the data from the database, and that is useful for the analysis as well as for the prediction. So this is a schema I developed for my database, right? And you can use it for any of the developing any of the databases. This is the format. So if you see the left side, I use the same format: number, protein, PDB, mutation, TM. Okay, the same format. If you see, you can see we can map exactly the same. Right? So I use the same for all the entries in the database. So in this case, you can easily write a code to see to retrieve data for whether TM or delta G and so on. So now this is the search conditions. So I have the various search options available. So you can use any of the options to search the data in the database, right? Either you can have the wild type data or the mutation data or the mutation present in secondary structures, helix, strand, file, right? Any data you want, so you can get from this database. So these are results. Okay, here I put lysosome. Lysosome is here, and we have the mutation here. Okay, this is E is mutated to H, and here you can see they changed the thermal stability of 0.1 degree. This is 0.9, this is 0.8. Right? This is the actual temperature, melting temperature. This is a change upon mutation. So if you go to this paper, you get all the details, right? This is the major aspect of this characteristics of database. Okay. Now, how we develop these biological databases, right? So in this case, mainly the biological databases are the relational database, right? Mainly the IBM, they developed this relational database. And in this relational database, right, it is the one in which the data are organized as tables. Okay, for each content, right? They are organized as tables, right? And these tables comprise several records called these fields. I'll show you the tables and the fields. This is called attributes. And this allows the later data to be linked to each other. So for any query, we don't have to put everything in one table. If we have the different tables, right, they can use the various operations, and then they collect the data from here and there and fetch the data for your query, right? So here I give the table 1. So here this is table 1 is here, and the table 2 is here, right? So here this amino acid is the full name, and this is 3-letter code, and this is the single letter code. This is the volume, and here is surface area, and this is distal group, whether the methyl group or guanidine group, amide group, and so on. So I have one question. Okay, so this is first question, very simple question. What are the 3-letter codes of the amino acids which have distal carboxyl group? Right? Can you give the answer for this? So just check the table 1, and what is the answer for this first question? What are the 3-letter codes of amino acids which have distal carboxyl group? Look at the table 1 and give the answer. What's the answer? Tell me quickly, I have only 15 minutes. I got so many slides to show. What's the answer for that? What are the 3-letter codes of amino acids which have distal carboxyl group from table 1? Am I audible?
Yes, sir.
Okay, then give the answer. This is a very simple question. Just you can look at the table and then give the answer. Very good. Aspen glue, very good. Yeah, just a carboxyl group is here. So go with that one. So this is a distal group, and I have the 3-letter codes. This is the 3-letter codes is here. So for this one, this is the asp, and this is the carboxyl here, another carboxyl here. Glue. Very good. So this answer. Okay. Now the second one, I got another question. Second one. So here I asked the 3-letter codes, but I got one more condition. I put the volume more than 125 angstrom cube. So what is the answer for this? Quickly you can write, because among the two, which one is answer for this? Here you have to look at the volume as well. Volume should be more than 125. What is the answer? This is more than 125. This is more than 125. Blue, right? So correct. So this is fine. So now fine. So this is correct. So glutamic acid is answer because this is more than 125. So now the next question. This is interesting question. So what are 3-letter codes of the amino acids which can serve as hydrogen donors? The question is, I have the amino 3-letter code, right, which have hydrogen donors. Can we get the information from table 1? Just from table 1, can we get the information? Yes or no? Can we get the information from table 1? No idea? No. Okay. I got this interesting. I got both the answers. Yeah. So somebody wrote yes, somebody is no. Okay. So because here we have only this information. There is no information regarding the donors. But donor information available in table 2. Right? So if we have only table 1, we cannot get the answers because this hydrogen donor information is not available in table 1. If table 2, we have the hydrogen donor information, but there is no information available for the 3-letter codes. So we cannot get the data answer from table 2 as well. In this case, you need to join, right? Go to the hydrogen donor, yes, and then get the carbon distal group. And based on the distal group, check the table 1 and you get the 3-letter codes, right? So in this case, it is a natural join. So now we can join table 1, table 2. So now this is the group, and here donor accept information, yes and no. I combine from the table 2, this from table 1, this is from table 2. Now we can use it. If it is yes, right, go to the group. Now from the group, right, you can get the 3-letter codes of the amino acids. So you can combine together. Okay, fine. So now if you go to the complex queries, for example, here there are so many. Donor should be there, but not acceptor, and surface area is in this range, and so on. In this case, it is difficult to check in the table. So we have used a structured query language, SQL language, right? This is syntax. Whenever you click on one particular column, like search option, it will select. It will create this SQL language, right? So we need a 3-letter code. We select from that table, amino acid table 1. Where these are conditions? Hydrogen donor is yes, acceptor is no, surface is more than 120, and distal group is method, right? Or these things, then automatically it will provide the answer, right? This is the thing. Okay. So there are various databases available in the literature. The Nucleic Acid Research Journal, the current impact factor is around 17, right? 16.9, right? It publishes article every January, first issue, collect the various databases and they publish in the journal and they maintain a database, right? This is the list of database in this website. This is A. So you put BCD and all these things. If it's a database, that is fine. right? So if you maintain all the databases, they listed it and they grouped into various classifications. And whatever the data you are interested in, just you can go to the website and you can get the various classifications and you can get the data right on particular classes. And every first issue of January, so in this website, this journal website, and you can get the journal which describe the explanation of all the databases published in the journal. Okay, so there are various database categories. So we have the nucleotide sequence categories and the protein sequence database and structure database, thermodynamic database. And if you see my database is listed under thermodynamics, right? And we have the database for the genomics, metabolomics, and so on, various databases, right? And PubMed is for literature, and that is the other molecular biology database. Fine. Okay, so now I show about the mutations. I talk about the mutations, and there are various databases available in literature. And we also developed some databases for the mutations and the disease-causing mutations. Okay, one is the HUVR base, that is for the human variant database, right? So here, as I told you earlier, so this is the schema. HUVR base is here, and is connected with various other databases. We get the experimental data from different resources. Because these resources are they have different information, they are not uniform. And we collected the data and we checked the literature and we they curated the data and put in our database. And we gave additionally all this information we gave in our database, right? So mainly two different aspects. One is at the gene level and the protein level, right? And another aspect is at the disease level. Okay, this is the structure level and this is the disease level. We give all the database information. Like there is the which disease, what the disease class, right? And the protein level, we have the what are the mutations, what uniprot ID, PDB ID, and so on. All the information we give in this database. Okay, show you the database now. Okay, here this is our website, right? And if you see the function and if you see the missense mutation, right? So if you do it, right? So it show all the data here. There's various options, right? We can retrieve the data. We have the link to the uniprot. Right, and you can have this analysis of the disease and the structures or the sequences and so on. Okay, this is another database called MutaHTP. This is for the mutations in transmembrane proteins, right? So you can see this is in this website, right? We focus the human transmembrane proteins, right? All the mutations. There's about 5,000 proteins in the human proteome. So here, this is our main database called HUVR base. And we use different aspects. There is a database and curation and the construct database and the feature extraction. So these are the information we calculated right from the available other resources, right? And this data we collected, right? So we have this information regarding conservation and the structures and topology and so on. And we have a lot of potential applications. We can have the search options, display options, and you can visualize the data, and you can download the data and use the data for the prediction as well as for the analysis. Okay, here this is. Sorry. Okay, so this is the overview of our database, right? So if you go down, so here you have search options. You can search with any of these conditions. So here you have the display options. Which data do you want into display in the output, right? So here this is your output, what it looks like, right? So this is your gene name. This is your nucleotide mutation. This is your protein mutation. And this is your uniprot ID. This is PDB ID. And this is the disease type, for example, this is a cancer and so on, right? So different things. So this is compare the database. Our database, we have almost several different informations, right? So you can use this website to get this MutaHTP or just to type my name. So you get to my lab website, right? And if there is a link for the databases, and if you click the database, you will get all the databases what you developed in our database, in our lab. Okay, so now there are various other databases, mainly for the cancer. These two databases are widely used for the cancer mutations, right? One is the Cosmic. So this is the biggest repository for cancer mutations, right? The catalog of somatic mutations, right? And they're commonly used in various analyses. And another database, this is the Cosmic. Another one is the TCG. This is mainly the DNA level, right? So cancer gene atlas. This is also widely used in literature, and you can see what will happen if you mutate a particular residue or nucleotide to nucleotide with respect to the disease or not. Right? These are based on the patient data. So they have the patients and they get the genome and they identify the mutation and they record it in this database. Okay, so here there are two different types of mutations. One is a driver mutation, another one is a neutral mutation. So if you see here, some of them are green, These are the neutral mutations, and some of them are red. Okay, some of them are red here. These are the driver mutations. Right? This cause disease. This is disease causing, and the passenger is the neutral mutation. So disease mutation, there is a contribute to transformation of normal cell through the cancer cell, right, and the tumor progression. And the neutral ones, that it have the changes but not occurred during the growth of cancer. So this is called neutral as well as the disease causing mutations. This is one example I showed here, one protein. So some disease mutations, some neutral mutations, right? So these are the common genes, right, which have the different types of cancer types, right? And if you look into the details, so there are some residues are very frequently present in these disease mutations, mainly arginine. Arginine is very highly dominant in the case of these disease mutations compared to the neutral mutations. And you can see different types of diseases. This is very dominant. Then the next one is, okay, arginine is highly dominant, that is fine. So arginine to which residue? For example, if you see arginine is very dominant, but mainly it is mutated to some specific residues like cysteine, okay, and you can see this is the histidine and arginine to glutamin Q. So right, then this is also there. Okay, this is 3.755. So there are some cases the mutation is highly prevalent, right? And some cases it is not. For example, the E is negative charge residue and this is a positive charge residue. This is the highly dominant in the case of the driver mutations. So you can do the analysis, and if these mutations, if you see they are there, then it mainly causes disease. Because if you use the neutral ones, there are some other mutations, not the same mutations. Values are very less, right? Some mutations which are also occurring in the case of this neutral mutation. And based on this odds ratio, you can see which one is dominant in the disease, which is dominant neutral. Some mutations are common in both because there is not for diseases, they are for the structure and function. So there is common in some cases. Right? So now we have the different cancer types. Right, we have the different types of mutations. Right, I think like this you can get the information from my papers. Right, and this I'll skip this one. And then we analyze, okay, what is the distribution of these mutations? So we found that these are the different hotspot residues. That means experimentally known to be disease causing. This has two aspects. One is this mutation you can see in various patient samples, as well as they did the experiments in the lab and then see what happened to these mutations. And they also found that this altered the function as well as this is highly for the disease causing. So when you get the distribution of this mutation, you get two mutations here, this three mutations here. Mainly if you see the disease mutations are clustered together. It is not spread here and there. Most of them are clustered together because we use a different line. Right? If you see most of them are clustered together, right? Then it is highly random. So if you see the values, so we can see the complete driver to driver. So its complete driver is very high. For this passenger, you can see the passenger to passenger is very high. So they want to be clustered together, all the driver mutations as well as the neutral mutations. They are also clustered together in the protein sequence. Right? Okay, so this I skip now. So now I'll go to the prediction aspect, right? So I'll show you how to discriminate, because now AI is very popular and we have the various machine learning techniques, right? So we can develop computational tools, right, for identifying these disease causing mutations. So initially we took the EGFR, the simple set of data, and then we extended to a huge data set. Right? Here there are 600 mutations from Cosmic and 266 driver and 334 passenger. And if you see it here, green are the passenger and red are the drivers. Right? And if you do the analysis, you can see 78% are mainly the intracellular region, right? And the driver mutations are mainly strand and coil. And most of the cases, there's the glycine and valine substitutions highly predominant in the case of these disease-causing mutations. Right? Then we did the experiments, right? So we collected data from the experiment. There are also functional assays. They also correlate this criteria, right, very well. So you can see one example here. So this is the EGFR structures, G791S. Right? This is your serine, and here this is your glycine, right? So this substitution of the glycine to serine, they destabilize the kinase domain by distorting the structural changes because they change in the hydrophobic interactions, right, in the wild type structure. Then if you see the experimental data of the disease, so disturbing the autoinhibitor interactions, that is the reason it's causing this cancer. So this is, you can see both this structural level as well as the functional level, as well as the disease level, you can see the correlation for specific these mutations. Then we developed a method. So first we collect the sequence data and then make the features and do the feature selection, and you can do the model using the machine learning or AI. So then evaluate the performance and then optimize the model, going the better performance. So various features we use, there are physical properties like molecular weight or size or shape, and we have the chemical properties like hydrophobicity, polarity, and so on. And we have the energetic properties. And for any property, for example, if the hydrophobicity of, you can say, valine is 2, for example, and hydrophobicity of this glycine is take 0, then delta P you can calculate. For example, if you see the delta P of, for example, delta P of valine to glycine, right, or the glycine to valine, right? So this value is P mutant. So mutant value is 2 because this valine is 2 minus glycine is 0, that is equal to 0. So for any mutation, you can calculate delta P. From this, you can see the other side, disease or neutral. You can relate if there is some specific values, this is disease. For some specific cutoff values, it is neutral, right? You can distinguish. So we have the program called Seek to Feature. Okay, this you can see in this paper, and we developed this software. So this will calculate the property values. For any property, you can calculate the average property value, right? Any property value. So this is another way. So I just explain now. So delta P mutation. So this mutation of SNP, that is for the mutant and the wild type. If this is 2 and 2, this is equal to 0, right? Depending upon the values. Right? Likewise, we have the DNA. So we have the property values for the DNA as well, right? So we can have these denucleated values, and you can get the average property values for the DNA as well. I just skip this because I don't have much time. So we have the server. So just we give the sequence in FASTA format, right? And here we have the mutation, and we have the different values, and we get the values from this one, and you can use it, right, for the purposes. All the properties you can get from this website, right? This is our website, and you can use these property values, right, for the understanding the properties which are important for the structure or important for this function or disease. So we have some nucleic acid based properties. So these are various properties based on nucleic acids, ACKT content, GC content, and so on. Right? You can get these numbers, right, as well from this website. Right? Okay. So I got one question now. So I discussed about the average property values, right? So if the property, take the property values, for example, if you we have the mutating alanine to alanine to leucine. The value is leucine equal to 3, valence alanine equal to 1. What is the P delta P? Delta P equal to, what is the value? P meter A to L. L is 3 and A is 1. What is the delta P? The mutant minus wild. What is the value for the mutant? This is wild type. This is mutant. So what is the value for mutant? What is the value for L? This is the data. 3, very good. 3 minus, what is the value for A? 1. Oh, very good. So this is equal to 2. Fine. So now we have delta P. Now we can say this is experimentally, we know this is disease or this is neutral. Now we get these numbers and we classify based any cutoff disease or neutral. It's not very simple, but I'll show you what is the performance we get. Okay, this is another our software, so we can derive our structure-based features. Okay, I'll skip this because I don't think I have time now. So we use machine learning techniques, right? Whether use the fitting the experimental data with many various features. The common one is neural networks. This is the several hidden layers. They develop this artificial neural this AI, right? So either we use neural networks or we can see the support vector machines to define this hyperplane so that it can separate the positives and negatives, right? They can classify whether it is neutral or this is disease causing. So now we can make it as the assess the performance, right? Using sensitivity, specificity, and accuracy, right? Which is positive and predicted as positive, experimentally positive, predicted positive, true positive. If experimentally it is positive and negative, and predicted is positive, this false positives, and experimental is negative and predicted negative is true negative. Experimental positive, predicted negative is false negatives, right? TP is true positive, F is false positive, T and true negative, Fn false negative. For example, here if you take B as positive, right, B as positive and N as negative, right? How many true positives? Can you tell the number? True positives means experimentally B and predicted also B. B and B. That is called true positive. Predicted is positive, experiment is positive. How many true positives here? What should be B? One, two, three. How many true positives? 12, 3, 4, then here 5. This 5, right? There is no B and B. What should be B? Right? That is 5. How many false positives? False positives means experimentally N and the predicted B. Experimentally N and the predicted as B. Wherever the N is predicted as B, that is false positive. How many false positives? Here 1, 2, 3. Anybody got the number? N and B? Four, 5, 6. Okay. False positive is 6. Okay. True negative you can calculate. Two negatives is both are N. Both the first line and second line, if you have the N, that's got true negatives. How many true negatives? 1, 2, 3, 4, 5. Can you count and tell the numbers? True negatives? Okay, calculate well. Okay. False negatives, right? False negatives means, right, you have the experimental is positive, experimental is B, and the predicted is N. So this is called the false negatives, right? So this is 1, 2, 3, 4, 5, 6. Okay, this is 6. Okay. So you can get these numbers, and based on that, you can calculate sensitivity, specificity, and accuracy, right? So you can calculate the sensitivity, specificity, as well as the accuracy, right? Sensitivity you can calculate using the formula true positive by true positive plus false negative. Specificity you can calculate true negative by true negative plus false positive. The accuracy, right? You can see the true positive plus true negative divided by true positive plus true negative plus false positive false negative. That means this is the correctly predicted. This is the total number of instances. This will give you the accuracy, right? Okay. So now I will give you a question. Right? We have the 100 residues, 95 in one group and 5 in another group. Okay. So out of 100, we have 100 cases. Okay. And 95 binding and 5 NB. The prediction method predicts all the 100 as binding. What is accuracy? So we have 100 cases, 100 residues, 95 as B and 5 as NB. And the program predicts all the 100 as B. What is the percentage accuracy? What is the answer? Okay, I think everybody is sleeping. Okay, I'll go ahead. Okay, yeah. So this out of 100, 95 is correctly predicted means accuracy is 95, but you can see sensitivity is 95 and specificity is 0. So in this case, this method is not a good method. I think I'll stop very soon. Don't worry. Yeah, because I don't think it is worth to spend much time on that. Fine. Okay, so I'll stop here. I think I'll take any questions or I think I'll stop. Okay, I'll stop here, and if you have any questions, just you ask me. I don't think it is good to proceed. Yeah. So if you have more information, just you take my books and more information available, right, in my books. Okay. Thanks for your attention.